Excellent. So now we're heading into our very last presentation of the 2024 workshop. We'll have Daniel Marrama, who presented yesterday, for those of you who joined us, talk about a collaboration that's been ongoing. So it's IEDB, UniProt and Protein Ontology, a collaboration. Thank you so much, Dan.
Thanks, Nina. Thank you, everyone, for joining. And if you've stuck through all 3 days, you're awesome. Thanks for sticking with us. So yeah, I'm going to be going over a collaboration that's been ongoing, as Nina mentioned, between us and UniProt and Pro and IPTMNet regarding our protein data. So in the IEDB, we aggregate protein data because when we have epitopes that come from what are called the same protein, they actually aren't from the same exact protein most of the time. So an author in a paper will specify a protein, and it's not necessarily the same protein between two publications. So it's kind of a challenge to capture all this data and to represent it that's intuitive for users, so that they can use it more intuitively and more efficiently. So there's 3 levels that we sort of aggregate the protein data. There's the curation level, there's the collation level, which is aggregation of these isoforms, and then there's the collaboration, which is taking other external resources and pulling them all in. So on the curation side, when a curator is going through a paper, they will often have to select a protein that is not necessarily specified by a accession ID. So in this example with hemagglutinin from influenza A virus, you'll see that there's like 3 different records from different epitopes, but they are not necessarily all coming from the same, in this case, genpeptides. So the author might sometimes just give a sequence, or it might just say it's from hemagglutinin and not necessarily give us a direct accession. So one of the things we have to make sure of is that the epitopes actually is 100% contained within the ID that we specify here as the source. So there are a lot of different possible accessions, especially for viral proteins. So more visually, what I mean is, let's say you have 3 different influenza A strains, and they all call them hemagglutinin within the paper, but let's say they all specify a different ID. So this represents a challenge because we don't want to just display every single hemagglutinin protein to users, as that would be just too chaotic and messy. And if you just even search hemagglutinin influenza A on UniProt, you'll see that there's a ton of records to choose from. And so we want to make this easy for users to just select hemagglutinin and see which epitopes they come from. So yeah, so it's more of a condensing that we do on our side, on the IDB side, where we select a representative hemagglutinin for influenza A. And we do this for all species and all proteins for each species. So the IDB has about 1,400 unique species, and there are hundreds of thousands of proteins that we curated. So we have to do this for all of them. And so yeah, what we call the protein tree on the IDB tries to solve this problem. And so when you go on IDB and you search our data via the tree, or the molecule finder, you'll see that for influenza A virus, for example, you'll not just see every single hemagglutinin that we have. You'll just see one representative entry, so that you can click on it and then search for all the epitopes that are contained within hemagglutinin. And when you also go to the antigen tab, when you search the table, you'll see that all these influenza A proteins just have one entry for each unique protein. It's not every single one. And so one other thing too is that we want to make it so that you can just search hemagglutinin, but you can also search synonyms of, let's say, hemagglutinin or any protein for example. So if you go to our molecule finder, you'll see that even, I don't know if you can see it here, but even for hemagglutinin, there's like HAEM spelling of hemagglutinin instead of HEM hemagglutinin spelling. So and you can imagine that there are many, many different synonyms for all the proteins that we have. So we want to be able to make it so that users can search that way. So as an overview of how we do this, so for each species that we have, a proteome is selected based on a number of factors, and these proteomes come from UniProt. And then all the source antigens that I mentioned that come from the paper that we curate are then aligned using BLAST for that selected proteome. And then we get the top hit associated with that, that BLAST alignment. And then we get the gene associated with it because we want to check different isoforms potentially, as maybe that the epitopes that we curate from are not necessarily found in the exact BLAST alignment, but they might be found in the isoforms associated with that gene as well. So we search the epitopes within all the gene isoforms using our PEPMatch tool, and then we might assign the epitope to a different isoform instead of the BLAST alignment and this is what we call the parent protein. And I'll give an example in a minute of how this looks like overall. So yeah, when you search on IEDB, you'll see in the antigens tab, what we call the parent protein is what you're seeing. And it's what this assignment is doing. So to begin with proteome selection, like I said, all proteomes are taken from UniProt. And we do this because UniProt has a nice curation process that they handle for proteomes, that they bring proteins together for different species. And there are 5 types that they define. And we basically, for each species, go through the list of each type and see if each proteome type is there for that species, and we select the best one. And then if there are ties, then we will search all the epitopes for that species and select the proteome that has the most matches for all of the epitopes. So the 5 types there's one that's representative. So this is actually basically doing the work for us. There's one proteome that is selected by the UniProt team to represent a species. So we just choose that one. Then there are reference proteomes. So a species can have multiple reference proteomes. For example, with E. coli bacteria, there might be many different strains of E. coli that are studied within a lab setting, and they're highly annotated proteins or proteomes. And those can be used as what they call a reference proteome. And so there might be many, and we might have to search all of them to see which one's the best one for our epitope data. And then there are what they call non-redundant proteomes. And these are proteomes that stand out as highly highly non-similar to other proteomes within UniProt database. So they are ones that basically have more diversity amongst the proteome sequences. And so that we might want to capture more that diversity so that we can capture more epitope matches. And then the last 2, we have what they call just an other proteome, which is not really annotated in any way. It's just a computational mapping from the genome. And the last one is not even really defined by UniProt, but something that we call as orphans, because within the UniProt knowledge base, there might not be an official proteome file or like collection, but there might be just a species taxon ID and random protein entries scattered amongst the UniProt knowledge base that we can just pull in and select from. So we do this hierarchy step through to select the best one. So after we've selected the best proteome, I'm going to show an example of how we do this assignment process for a given source antigen. So let's say we take a cow as our species, which is the scientific name is Bostaurus, and we have a protein curated from a paper. It's called, in this example, alpha-lactalbumin. and we have a bunch of epitopes associated with alpha-lactalbumin. So then what we do is we take that full sequence from the source antigen and we blast it against the selected proteome. And then we take the top protein hit, like I said, which is based on sequence identity, how much does it align? And most of the time, actually, it's will be like 100%. But you can imagine that if you have viral proteins that are slightly different between each strain, there might be like one residue off or so. So we take the top hit based on alignment. And then we take the gene of that top hit, and then we get all the isoforms associated with that gene, which will be in the selected proteome. So the nice thing about UniProt is that they will, in their proteomes, they'll often collect all the isoforms for a given gene into one, so we can use those. And obviously with like things like viruses or bacteria that don't do like alternative RNA splicing, this might not be as relevant, but we still do this process to see where we take all these isoforms and then we can search these epitopes within each isoform to see if they have an exact match in any one of them. And again, like oftentimes it'll just be the top hit that we select, all these epitopes will be found in. But then if not, we take the best isoform. And then, yeah, if there's no so there's no match in any of the isoforms, we just assign the epitope, the top protein hit via the blast alignment. And we call in this process the parent protein, like I mentioned. So one other thing we do to aggregate our data is we want to capture things like receptor antigens, so like MHC molecules, TCRs, BCRs, or immunoglobulins. And because oftentimes these are source antigens that are found for a particular species. So if a species is a vertebrate, which of course are the ones that produce these antigen receptors, we use a tool called ARC to basically check if the source antigen is in one of these categories or not. This is a sequence-based classifier that can tell us whether it's one of these, and then we can basically assign that source antigen one of these nodes, and that can also be searched in our protein tree. And it's kind of vital that we do this because UniProt does not handle these things well because of the extreme variability, of course, with these receptor antigens. And so in this example, alpha-lactalbumin is not an antigen, so we just skip this and we use the process that I talked about before. And so then, yeah, one other thing that we do is we often will use allergen nomenclature for particular allergens that have these IUAS, what we call WHO IUAS names. They are often in this format where there's like 3 letters and then 1 letter and then a number. And so alpha-lactalbumin happens to be an allergen that is highly studied. So any allergenists want to maybe search on the IEDB for alpha-lactalbumin, they might use BOSD4 instead of the official name. So we want to actually give this allergen that BOSD4 name. And then one last thing we do for the protein tree is capture the fragmenting of proteins. So oftentimes proteins will have like a signal peptide or transit peptide or other things, or propeptide that are have a biological function, and they might often have some immunological significance as well. So we want to capture these segments of these, fragments of these proteins, and display them on the tree. So in this example, alpha-lactalbumin or BOSD4 has a signal peptide at residues 1 through 19, and then the rest is the mature protein. And so the signal peptide is cleaved off often in proteins that have signal peptides are often that section is cleaved off, and it might be of biological or immunological significance. And so we want to display that, and we only display these fragments that actually have epitopes contained within them. So you can search based on these fragments themselves for epitopes that are contained. And yeah, we take this from this information or this data from UniProt and that's how we populate it. So just to demonstrate how this looks, so you would go on the front page of the IEDB, as you've seen before. You'd select in this antigen box here, and you could search by the protein name or the protein ID, or you can filter by the species. And then you'll have like let's say in this example, we're searching for cow proteins. You'll have the molecule finder at the bottom here, giving you the protein IDs here and the names, and then the synonyms. So you can search by synonym, you can search by the actual name itself, you can search by the ID, and then you can click this text highlighter, which will actually bring you to the entry of that you want to look at. You can search by that way. So moving on now to the collaboration side of this presentation. So we, like I said, have been having an ongoing collaboration with Pro group, the UniProt and IPTMNet, to enable functionality for our data. So after we've aggregated these proteins into one representative, then we want to expand in a way our data so that we capture other things that are relevant. So somebody might be interested in one particular protein, and they want to know more about that protein that is captured by Pro or UniProt or IPTMNet. So for Pro, they have ontological representations of these proteins that then give an explicit representation of different proteoforms. And the UniProt, of course, has many different annotations and metadata for each protein that's within their database. And for IPTMNet, that's a tool that and database that captures the post-translational modifications of these proteins at particular positions and within biological contexts. And so here's what Pro looks like. So you have like hemagglutinin, and you have a bunch of proteoforms here. So this data might be relevant. And then the UniProt data, we have like subcellular location or the sequence or the domains or the structure and many other things, or the function even. And then the IPTMNet, like I said, we have post-translational modifications at particular sites. And there's a bunch of other metadata around that, like the PMIDs, where this evidence comes from, or in the context of certain diseases and things like that. And also these link back to Pro. So these are kind of an interconnected resources that we want to capture. And so as of right now, we have developed what we call antigen summary pages that are now live, and they capture a bunch of data for each of these parent proteins. And you can see here, you have the parent protein ID for hemagglutinin, all the synonyms associated with that protein, all the epitopes associated with it, and the publications, the assays, the 3D structures, and then the hosts that were these epitopes were tested in, and the diseases that they were also within that context of the assays. So this is like, if you want to basically study a particular protein, this is very useful to see a snapshot or an overview of the protein itself. And what we're also trying to do is link out from the resources I just mentioned to capture annotations like the gene name or function or structure, things like that, or post-translational modifications, so that we can display that on the IEDB or link them out so that users can more easily streamline what they're trying to find. And so a part of this collaboration also was useful for us as we've improved our data based on things that we found, like protein IDs that were no longer valid, or an organism was assigned wrong based on our curation or the paper, or post-translational modifications that just were not valid or just impossible. So that we've been able to update and improve our data based on this collaboration as well, and enforce new rules for the future so that we can catch these things as they come in. And so the last thing we have is plans for linking out to these various databases. We already do have particular links to Pro in particular, and on these engine summary pages, but we want to also link out to everything else. And we also want to eventually capture certain annotations, like whether the protein is a cancer antigen or an allergen or an autoantigen. And we can give these based on weight based evidence, like the quality of data that the assays come from, or the paper and things like that. So we're going to be working on that. And then everything I talked about in this presentation had to do with proteins, but there are also, of course, non-peptidic entities that we capture in the IEDB that we want to also include for our summary pages. So, yeah, that's it for me. Thank you, guys.
Wonderful. Thank you so much, Dan. Much appreciated for ending the day on a high with a collaboration that we've been working on for some time. So, and to the point, I'm about to post it on Twitter or X, but we do pride ourselves in making our data fair, so findable, accessible, interoperable, and reusable. And these collaborations are just one way we're able to do this, just, you know, to make the data fair for all of our users, capital FAIR. But now we'll move into the last question and answer section for the day and for the year. So I'd like to introduce Jennifer back with Dan to kick off the last Q&A. So Jennifer, if you'd like to take the first question.
Okay, sure. And I just wanted to say there are a few questions. We actually got a lot. Thank you. There are a few questions that were relatively specific that I felt like the audience might not all benefit from hearing the response, especially if I can only pick a couple of questions to answer. So I tried to answer those in text. You should be able to see them. If you cannot, then please email me at immuno@lanl.gov, and we could talk about it further. But I did try to answer them through text, but I'll answer a few live. So the first one is, so waning CTL and humoral response is a characteristic of HIV infection in the long term. And I would say yes, absolutely. So CTL exhaustion definitely happens in response to HIV infection, because you just get continuous antigen presentation that's initially exciting T cells, and this is resulting in exhaustion. So this is really well studied. There have been, I think, even clinical trials trying to use immune checkpoint blockade inhibitors like PD-1 inhibitors to kind of restore some of the CTL response to HIV infection. And I think those have been, at least somewhat successful. Like whether they're going to be cost effective to do in sub-Saharan Africa, that's a totally different question. But this is definitely a strategy that's being tried to just kind of restore, full CTL responsiveness to HIV. I haven't heard so much about waning antibody responses. I think that those tend to continue a little bit more robustly in HIV infection. You still are getting somatic hypermutation. It's possible you're instead just getting antibodies going kind of down the wrong path, especially as they continue to do somatic hypermutation after many years of HIV. But yeah, absolutely, CTL exhaustion is a pretty well-known thing to happen in HIV, and it's definitely a problem.
Yeah, so there's a question about how we handle canonical form of the protein versus isoforms with UniProt numbers. So yes, this is a big problem actually, in a way, where UniProt will sometimes have isoforms where there'll be the canonical version, and then they'll add like a number to the ID as another isoform, but then they will also include isoforms with a totally different ID. So often what's nice about UniProt, though, is that they will capture all of those isoforms still within the proteome. And so they are all kind of given a chance for this alignment that we do for BLAST. And whatever comes out as the best hit is what we select, whether it's the one with the number or one with a totally different ID. And the ones obviously with the numbers, like under the canonical version, have more annotations. So they're usually better to pick if possible. And we do have some preference for those ones that have better annotations.
Okay, so I'll take the next question. So this is an interesting one. Can an individual be infected with multiple strains of HIV, or can HIV mutate within the host rapidly enough to outweigh the antibodies initially produced? And infection with multiple strains of HIV does happen. I think it's relatively rare. I mean, I think most individuals that have HIV were just infected with one strain initially. If you are co-infected with multiple strains, I think it's actually advantageous. You're more likely to develop broadly neutralizing antibodies, probably just because you have a lot more HIV diversity to start with early on in the infection. But in most individuals, the antibody responses are occurring to just a single strain of HIV the individual is infected with, and the HIV strain they're infected with just is continuously mutating within the host, and you're getting antibody responses to that. The individual doesn't need to just keep getting infected with different strains of HIV in order to, mount evolving antibody responses. Yeah, I would say most individuals are just infected with one strain. And HIV infection is actually relatively rare. I mean, in most encounters, like you're not going to become infected with HIV. So I think being infected with many different strains is actually like a pretty rare phenomenon.
All right, I'll take the next one. The question is about UniProt having unreviewed proteins and how we sometimes use those on the IEDB, despite there's like an official one or a SwissProt one. How do we keep track of these? So yeah, there are a bunch of Tremble entries that we still use, probably even more than SwissProt ones, unfortunately. It's just given the size of the UniProt KB and the percentage of Tremble entries is, I think, around 80% or maybe even more. And when we do that alignment, we take the best hit, and oftentimes that might be a Tremble entry. And so we still want to capture those if it's a better hit, because the epitopes that we contained within the source antigen will be captured within that best hit. So it's unfortunate in a way, but it's kind of just the nature of how UniProt just annotates their data right now.
Okay, so I'll take the question, is it possible to check if immune responses we see following vaccination are cross-reactive, i.e. epitopes seen in other infections? So in our database, no. So we really only collect HIV information. We have a little bit about like simian immunodeficiency viruses that are closely related to HIV, but we don't curate data on other types of viruses. There are definitely things like cross-reactive epitopes in HIV and say CMV. There might be information in the HIV record in our database about it being cross-reactive with CMV, but you're not able to like search for say like CMV reactive epitopes. I mean, this is, I think, a great reason to go to IEDB, type the epitope in, and you might see that it actually is cross-reactive with another virus or maybe like gut bacteria or something like that. Yeah, I would say go to IEDB.
All right, the question is about ARC. Can it also recognize non-classical MHC molecules like CD1 isoforms? It's a really good question. It does not right now. It does capture like it does predict for between MHC1 and MHC2, and sometimes even down to the allele. If it's, I actually don't exactly sure how it grabs the specific one, but sometimes it does have more granularity, but it does not capture, it does not recognize things like CD1 isoforms and stuff. But there is a ARC 2.0 that we're working on. So frankly, I'm not privy to how that's being developed. So possibly. But also one more thing about ARC is that it is on our GitHub. So if you go to the IEDB GitHub page, ARC is open source, so you can use it.
Okay, so we received a question. Do Lenal tools provide sequence data for both antibody and antigen side, being both epitope and paratope sequences? So the answer is kind of. I mean, we definitely have antibody sequences for some antibodies in our database. You can download like all the data in our catnap neutralization database for the antibody sequences we have. We don't have them for every antibody, but we have them for some of them. We don't currently have anything for paratopes. I mean, we've thought about making like a paratope database. We haven't done it yet. I don't think it's anything we're going to do in like the next year. It's not impossible. We eventually do it, but no, unfortunately, we don't have anything for paratopes.
All right. Do you have an example of where a signal peptide or propeptide is immunological relevant? Yeah. So I mean, the example I showed with BOSD4, there's a signal peptide fragment, and there are epitopes contained within that. So I mean, technically that would be immunologically relevant, but things like insulin, where the C-peptide is released into the bloodstream, and in type 1 diabetes, I'm not sure if C-peptide is recognized or other A or B chains, but there are a bunch of different examples in our data, and you can search actually by signal peptide in our data using the molecule finder to see which epitopes are contained for each one of those.
Okay, so I have a question. If I have my own antibody, is there a way to compare it with the antibodies in our database? Can I do the same analysis, being catnap, while adding my own antibody? And the answer is yes. You ideally would have antibody neutralization data, and some of the viruses tested would be the same, as the viruses we have tested in our database. Yeah, you can add your own data. You actually don't have to have neutralization assay data. If you have any kind of numerical data that you score in some way, you can put it into the catnap tool if you also have a virus sequence to go along with it. Yeah, we don't have a way, like if you have your own antibody and you say, I know it neutralizes these 3 viruses, can you predict what other viruses it's going to neutralize? We don't have tools like that. But yeah, there's a custom catnap. Hopefully it's relatively easy to find on our database website. If it isn't, or you have questions about using it, please just send us an email.
All right, this question is about PepMatch, which is not related to the presentation, but I'll still answer because I don't have any other questions on my end. So the question is about using PepMatch and how to generate your own pickle and database files for use. So I kind of reference you to our GitHub. We have the PepMatch tool with documentation on our GitHub, and there's also documentation for using the next-gen tool like I discussed yesterday. So you can use it either on our front end or locally using things like pip or Python library or even the command line. So if you're having trouble producing what you need, just either raise an issue on the GitHub or send me an email, and I'll help you out.
Okay, so another question is the source of the database clinical data? And yes, partially. So in the HIV immunology database, like nearly all the data we have is from publications. And some of these could be clinical publications. I mean, you of course have publications looking at people that are infected with HIV in more of a clinical sense. A lot of the data that was used in the publication is controlled data. We don't get access to stuff like that, but we collect data from publications for the most part. We occasionally get some other data that's submitted directly from users, but that's pretty rare. The HIV sequence database, we collect GenBank entries. I think sometimes people wonder, like, do we get sequences from health departments, from people that they have that are infected with HIV? And the answer is no. I mean, we generally don't, get set data like that, and we don't try to pull in data like that. We go from GenBank for the sequence database. Hopefully that answers your question.
Jennifer, if you want to answer the last one, I don't think there's any more from me.
Oh, sure.
Yeah.
Yeah, so this question asks, is there a tool for finding active site of HLA protein and identify anchor position of peptide binding to HLA and a position that would bind to a TCR? So for the first part, yes. For the second part, no. We have our motif scan tool, and I'm going to just kind of share my screen really quick. I just put the sample input in it. Our motif scan tool will show you HLA anchor motifs. So the second amino acid, so this is for class 1, the second amino acid is in blue and the 9th amino acid is in purple. So these would be considered anchor motifs for different HLAs. So this should make it easy to find anchor motifs for class 1. We don't have a tool that predicts binding to a TCR. We are considering adding HIV TCR data to our database in the next, maybe year or so, but we don't have any kind of tool like that currently. And then the last question is, do you have patent data? And I'm not sure if that was meant for Dan or I, but I would say the answer is probably no. I don't believe so.
Yeah, I'm not particularly sure what you mean by patent data, but I don't think so either.
Amazing. Thank you both so much. That was the last question in the chat, in the Q&A, sorry. It's been a long 3 days. But thank you, Jennifer. Thank you, Dan, for the great presentations and the great questions. And really a big thank you to everyone. If you've stayed till the end, the questions across all 3 days have been quite incredible and some of the most we've ever received. So that is much appreciated.